Hello, my name is David Roos, and I'm a professor at the University of Pennsylvania in Philadelphia. And welcome to the third in this series of iBio's lectures on my 5 favorite parasites, organisms in the phylum Apicomplexa, which include a wide range of pathogens responsible for diseases such as malaria and toxoplasmosis. And as you may have heard in the last couple of talks, we've discussed various aspects of the cell biology of these parasites and how they provide a fantastic window into a wide range of cell biological and molecular genetic studies on these organisms and a window onto eukaryotic pathogens in general. In this talk, I'd like to turn our attention to another aspect of technologically driven science. In fact, an area that's really changing the way we think about biology as a whole, something that I'm sure has been touched on in many of the iBio seminars. In particular, that's the area of genomics, an area which is characterized by vast amounts of data, complete data sets for all of the genes in the genome, all of the proteins that are expressed, and the interactions between them. And with the genome projects for humans and for many other organisms, such as the mosquitoes that carry malaria parasites, and from the malaria parasite itself, Plasmodium falciparum, with these large-scale datasets available, we can start to look at many interesting biological questions and to explore those in interesting ways. So by way of background, or to provide some sort of a context, let me remind you that in the last couple of lectures, we've discussed a remarkable cell biological system present in malaria parasites. And I've told you how malaria parasites and their relatives harbor a fascinating organelle which they stole from an ancestral plant or alga. When an ancient parasite, symbolized here by this cartoon diagram indicated in pink, ate a eukaryotic alga and retained the algal chloroplast as a distinctive organelle that is essential for parasite survival, although it is no longer photosynthetic. Here's a picture of that organelle, colored in green in an electron micrograph of a malaria parasite, quite distinct from the parasite nucleus or the Golgi apparatus and specialized secretory organelles involved in invasion, the inner membrane complex that we talked about in the first of this iBio lecture series that's critical for the assembly of parasites in a remarkable process, and the apicalplast, the apicomplexan plastid, surrounded by multiple membranes, distinct from a primary endosymbiotic organelle, the mitochondrion. And I told you how this organelle is essential for parasite survival, and we now have effectively a complete metabolic pathway map, including many enzymes and pathways which are the subject of intense interest as possible targets for new anti-malarial development. What I also told you in the last talk is that that metabolic pathway map was described using a variety of traditional biological approaches, biochemistry, cell biological purification, more modern approaches of proteomics, but that the most successful strategy for identifying pathways and processes associated with the apicoplast was in fact a bioinformatics approach, taking advantage of the availability of genome sequence data, some of it complete, some not, developing a range of computational and experimental screens to identify proteins that had one or another features that we suspected might be associated with the apicoplast, giving rise to a candidate series of genes which then be tested experimentally at the laboratory bench. So this is, I think, a dramatic illustration of the success of computational biological approaches for practical application in expediting traditional biological research. And it rapidly became clear that the same sorts of approaches that were so successful in asking questions like, find me plant chloroplast genes in malaria parasites, might readily be used to address a variety of other questions. We might want to ask, for example, can we find proteins on the left-hand end of chromosome 10 in malaria parasites which have multiple transmembrane domains and might therefore be involved in importing nutrients or exporting drugs and play an important role in drug sensitivity or drug resistance. And so the desire to be able to address questions like that gave rise to the Plasmodium Genome Database, which we will talk about further as part of the Plasmodium Genome project. PlasmoDB, the Plasmodium Genome Database, is accessible at plasmodb.org, and this project has itself been so successful that it's given rise to a series of other pathogen databases. Let's take just a moment to talk about the philosophy behind PlasmoDB. Our goal in this project is to take information that emerges from genome sequencing projects, ideally curated information, but to integrate that with automated analyses, recognizing that the rapid pace with which data emerges from these large-scale projects precludes the ability to look at each and every bit of information manually. We want to incorporate data that comes not only from genome sequencing projects and cDNA sequencing projects, but also information that comes from population genetic studies, from functional genomic studies on RNAs and proteins, from chromatin modifications, protein-protein interactome studies, clinical outcomes data, and many other sorts of projects. To provide that data in a rapid way that, most importantly of all, enables laboratory researchers like you, like myself, like individuals in my laboratory, to ask their own questions. What I mean by this is that our goal is not simply to provide a catalog, an encyclopedia of malaria, in which one could look up the answer to any question that might arise. And the reason for that is best manifested by the apicalplast project. So, for example, imagine that we had a catalog of all of the information associated with malaria parasites circa a decade ago, and we wanted to look up the answer to the question of find plant genes in malaria parasites. That's not an entry that we would have found in that encyclopedia. And yet, we're able to identify those genes in what seemed like a nonsensical question by taking advantage of generic tools that allow us to look for proteins that have particular attributes associated with targeting to this organelle or its evolutionary history, as we talked about in the last lecture in this series. And this project as a whole has been quite successful with worldwide access, many thousands of hits per day, from dozens and dozens of countries, over 100 countries around the world. Our motivating goal is that there is no such thing as a stupid question. So let's see what we would observe if we were to look at the Plasmodium Genome Database, and I encourage all of you to do so yourselves. So let's turn to look at what we would see if we look at the Plasmodium Genome Database, and I encourage each of you to do so yourselves, and we'll be spending a bit of time on this database. If you were to look at that, we can enter the database from the standpoint of an individual gene. This particular gene, a gene with the uninformative name PF10_0407, corresponds to a gene that is annotated as a dihydrolipoamide acetyltransferase, at least putatively. Now, this gene may be of interest to you. It's of no particular interest to me, we can see that it is on chromosome 10, located at about 840,000 plus nucleotides from one end, that it's the subject of an ongoing annotation effort, including modification of the initial gene model. It appears that a new exon has been added into this predicted gene. And I've excised a vast wealth of additional information that comes from expression profiling studies and computational analyses of predicted motifs and knockout studies that are indicated down at the bottom of this gene. And if you know more about this gene, I'd encourage you to click on the link for user comments, popping up a window, adding additional information that can serve as a global online laboratory notebook about this particular gene so that the information is available to others. Now, of course, you might not want to explore the genome from the standpoint of individual genes one at a time. You might want to look at the genome from the standpoint of the genome sequence itself. And so we can take a look at this from a chromosomal type of view. In this case, we're now looking at chromosome 11, at the entire 2 million nucleotides of chromosome 11 spanning this range across above. And there are dozens or hundreds of genes on this, too many packed in together too tightly to be able to explore. And we can look at additional information, in this case, displaying information related to polymorphic data coming from the emerging genome sequences of dozens and soon to be hundreds of malaria parasites, in this case color-coded to represent those that are coding versus non-coding, or synonymous versus non-synonymous polymorphisms. We can zoom in on that further and display additional information, in this case from a comparative genomic standpoint, looking at chromosome 4 from Plasmodium falciparum and comparing that genome sequence with comparable regions, synthetic regions, regions which can be aligned from Plasmodium vivax, another human malaria parasite, or Plasmodium yoellii, a rodent malaria parasite. And by aligning these, we can see that virtually all of the genes across this long span of DNA, hundreds of thousands of nucleotides, can be matched up one-to-one with genes in the Plasmodium vivax genome, with a few exceptions. There are some regions that don't line up very well. We seem to be unable to find a corresponding portion to the telomeric ends, the left or the right telomeric ends of this chromosome, and that's perhaps not surprising, given that this region is rich in polymorphic genes specific to Plasmodium falciparum. Similarly, we have other gaps. This gap here, in which more polymorphic antigens from Plasmodium falciparum correspond to a break between two contiguous sequences of Plasmodium vivax, and another region down here in which an insertion of several of these polymorphic genes absent from Plasmodium vivax corresponds to no obvious genes in Plasmodium vivax. And here, conversely, a region, a family of genes, Vir genes in Plasmodium vivax, which seem to have no match in Plasmodium falciparum. Interestingly, we can see that while the Plasmodium yoellii genome has not been assembled to completion, there are scores of individual contigs aligned here. We can nevertheless find a one-to-one mapping between most of these genes and the genes in Plasmodium vivax and Plasmodium falciparum. Thus, while the genome sequence for Plasmodium yoellii is not complete, it's sufficiently complete that we can find most of the genes that may be of interest. Now, I tried to make the point that these tools are not simply resources to look up the answer to these questions. How many genes are there on chromosome 4 in Plasmodium falciparum, but can be used as research tools as well. And I'd like to give you an illustration of that from an interesting study that came as a result of a workshop for East African students in Tanzania, who were excited by the recent release of Plasmodium vivax data and wanted to explore that a little bit differently, a little bit further. And in the course of looking at them, they noticed there are certain regions of the chromosome, and here a small portion of the Plasmodium falciparum chromosome, chromosome 4, zooming in on that portion of the larger chromosome that we looked at earlier, they noticed a particular region with no annotated genes that was extraordinarily rich in A and T nucleotides. The percent GC competition is virtually nil, even in the context of Plasmodium falciparum, the most AT-rich eukaryotic genome known. And so they asked the question whether this region, which is known to correspond to the centromeric region of the chromosome in Plasmodium falciparum, might also correspond to the centromeric regions in Plasmodium vivax or Plasmodium uelii. And so they pulled up individual tracks to display the genomes of Plasmodium uelii and found that in this case, the incompletely assembled Plasmodium uelii genome contained no sequence which was known to map to this region. But in Plasmodium vivax, we can see a single contiguous piece of DNA that spans this region is also poor in annotated genes. And sure enough, when these students turned on a display to visualize the A and T nucleotides, they see an AT-rich region of the Plasmodium vivax genome, which undoubtedly corresponds to the centromere, these genes as well. So just to be clear, a group of students with no previous exposure to these approaches sitting in Tanzania was able, for the first time anywhere in the world, to carry out exploratory research computationally identifying highly probable centromeric regions of the Plasmodium vivax genome. Now, this is certainly a question of great intellectual and academic interest, but when we're dealing with important human pathogens, such as malaria parasites, we obviously have many other concerns as well of practical nature. We might want to ask, for example, what are appropriate targets for drug or vaccine development? And in addressing those kinds of questions, it's worth taking a little bit of time to think about what the questions are that you'd like to ask and how to formulate those in computationally accessible form. Just as we asked for the apicalplast, find all genes that have one or another aspect of the targeting signals associated with proteins destined for that organelle, and one or another of the evolutionary signatures that suggests a plant or algal plastid ancestry. We might want to ask, if we were looking for targets for drug development, for parasite homologs of genes that are known to be targets for drug development. Dihydrofolate reductase, for example, a prominent target for chemotherapeutic drugs and indeed for drugs effective against malaria. Or proteases, a more recently explored class of targets for a variety of infectious diseases. Genes that might be associated with the cytoskeleton, that might highlight those remarkable aspects of parasite division that distinguish them from the way our cells divide that we talked about in the first iBio lecture in this series. We might want to look for proteins associated with the apicalplast or that are expressed at appropriate stages of the parasite life cycle or in the appropriate place or for which we have candidate inhibitors. All of these are questions that might be relevant to drug target identification, and we'll come back to look at some of those questions a little bit later on. But as maybe a more challenging exercise, I'd like to think instead about vaccine candidates, and this is, I would submit, a more challenging exercise for at least two reasons. First of all, because we know perhaps less about what constitutes an effective vaccine target. And secondly, because I, for one, claim no expert knowledge about vaccinology. But even as a naive immunologist, I might imagine that we might want to look for genes, for example, that were immunodominant or expressed on the surface of the parasite. Now, I'd be happy to take you through a series of graphic illustrations of how one might go about identifying candidate drug targets or vaccine targets. But I would urge all of you, when confronted with resources, online resources that are designed to be able to explore these projects, to challenge yourself and challenge whoever is introducing you to those resources to let you explore it on your own. And so rather than providing a series of of pre-prepared graphic illustrations, I'd like to run a live demonstration. We're going to take a break for just a moment, and I'm going to set up my laptop here to be able to run a live experiment, a series of questions that's designed to identify candidate vaccine targets. And I hope you'll find this of interest. It's something you may want to follow along on your own computer either now or at some other time. So let's take just a break and get this set up.